Good morning, ladies and gentlemen. My name is Lê Hà Phương. I'm a president doctor of Hà Nội Medical University Biochemical Department. Our presentation of today is about the plant-derived vaccines. My presentation has 6 parts: current context, advantages, historical timeline, procedures, challenges, and future prospects. We're going in the first part of my presentation, current context. As we all know, there are 3 ways to produce vaccines for human use. The first way is using the inactivated microbial pathogens in eggs or mammalian cells. The second one is RNA or DNA fragment from the pathogens, and the final one is the pathogens protein produced in mammalian cells or the insect cells that are purified for the vaccine formulations. The recombination vaccines offer a very high, particularly high safety, high stability, similarly high immunogenicity, but it requires complex and expensive productions, transportation, and storage processes. Here we got a table that we can see the advantages and disadvantages of the most common host cells. We got the mammalian cells, insect cells, bacteria, and yeast. As we all can see, the most popular host cells, we can say that the mammalian cells. The production of mammalian cells is quite expensive, take time to control, it's not easy to scale up. The human pathogens, contamination risks, and the heterologous product. The newest one is yeast. Also, it's quite difficult to describe the cell of yeast, and this similarly the capacity for the glycosylations. So we're going into the advantages of the plant-derived vaccines. The first one is cost effective. It's quite quick and cheap to grow. The crop protein are optimized. Large-scale protein productions and the capacity to produce a complex protein by performing many post-translational modifications, including the glycosylations and disulfide-bond formulations. No pathogenic contamination risks. Diversity of plant species used to be the whole system, and one of the most interesting things is the possibility of direct oral delivery. We are talking about the oral delivery of the drug candidate. The traditional biologics is quite expensive downstream processing and require a cold chain for storage and transportation, in addition to the sterile needle for their administration. But oral plant-derived vaccines will be protected from the acids and enzyme in the stomach via the plant cell wall, because the human digestive enzymes are incapable of breaking down the glycosidic bonds in carbohydrates. In the small intestine, the commensal microbes digest the cell wall and release the antigens. The antigens are filled with a suitable transmucosal adjustment, like CTB, can be delivered more efficiently to the immune or the circulatory system. In short, we got the transgenic plant vaccines can minimize the painful needle. There's no cold chain requirement, eliminate the costly and complex fermentation, and improve the cell life for years at ambient temperature. So we have to know more about the historical timeline. The first time that Stietro et al. Demonstrate for the first time that the plant cell have the capacity to produce a complex multimeric mammalian protein was in 1987, and until the 2006, the United States Department of Agriculture, the USDA, approved an animal health vaccine developed by a company in USA using tobacco NT1 suspension cells. And the newest one is in 2022, the COVID-19 vaccines produced in Nicotiana bentamiana has been approved in Canada Medical and Kentucky Bioprocessing Inc. in USA. Recently announced an FDA-approved COVID-LVP plant-derived vaccines against the COVID-19. So we are going in the main part of my presentation is the procedures. As we all know, there are 3 popular modalities of expressing the heterologous protein molecules in plant cells. The first way is stable nuclear expression, the transient expression using the non-replicative or viral vectors, and the final one, the newest one, is transplasmic expression within the chloroplast. We're going in the first way of this transportation is using the stable nuclear expression. The bacterium was used is Agrobacterium tumefaciens. The tDNA plasmid removed from the bacterium and cut using the restriction enzyme. The vaccine's fragment DNA cut with the same enzyme and added into the recombinant plasmid and added again into the bacterium. The tDNA carrying the inserted vaccine DNA incorporated into the plant cell chromosome and control to transform into a transgenic plant. Host plant species for this kind of transportation we can name like potato in 1998, rice in 2023, banana tomato in 2019, and go on. One of the most popular host cells for this kind of transportation is tobacco. It was the first time mentioned in 2007. The transient's nuclear transformation is quite similar to the stable one. It's including the inoculations, scale-up, transcirculations, injections, and control. In the same conditions and control process, there are less than 10% plasmid integrated into the chromosome and more than 90% exist independently like an extra chromosomal plasmid. Here we got a vacuum infiltrator construction and process in reality. Picture A and B, we can see the tree hold with the wings and dip into the infiltrated part and put into the chamber. The following picture, we can see the full flash of the complete vacuum infiltrator, the vacuum pump, connection from the vacuum pump to the vacuum reservoir. The 3 is vacuum reservoir installation valve. The 4th one is connection from the vacuum reservoir and the infiltrating chamber. The 5th is pressure guard. The 6th is the air intake valve, and the final one, the vacuum reservoir, is the 8th. The G picture is a representative lift from the one plant infiltrated with an expression construct from the green fluorescent protein after 5 days. Once in the cells, DNA molecules can be integrated into the chloroplast genome through the DNA recombination mechanisms. Currently, antigens production in chloroplast had advantages including the higher recombination protein productions, but there's no post-translational modification machinery in this kind of transportations. Here we got a table that we can see the expression plant species, the disease, and the method of transportation. Most popular expression plant species and the method of transportation is tobacco and their Agrobacterium. The disease is ranged from the colorectal cancer to the HPV or the Zika virus. So how can we extract the vaccines from the plant? We have to gather fundamental information about the target protein to the bioinformatic analysis software like the PL, cysteine content, protein stability, and go on. The available system for the biopharmaceutical manufacturing vary based on the plant type and the site of localizations of the protein products. For example, we got the MABs are typically segregated into the apoplastic space via the endoplasmic Golgi secretory system and subsequently recovered from the extracts of the plant material to the standard filtrations as well as the chromatographic approaches. Here we got a downstream processing method in molecular farming. We got the extraction, clarifications, purification. We can use the chromatography, membrane separations, genetic tasks, or other kind of transformation. And after drying and formulations, our vaccines are ready to use. We're going in the challenges. So a question is why a commercial plant-derived vaccines have not been made after 2 decades of plant vaccines development? The first reason that I think we have to care about is the selections of antigens and plant expression hosts. The second one is dosage consistency. The third one is vaccine manufacturing according to the GMP procedures. And the final one is one of the most important things that the accreditation of several health organizations such as the ICH, the WHO, EMA, or FDA. Although there's a lot of challenges in this kind of vaccines, it also have a lot of future prospects. The first kind of vaccines in future prospect I want to mention about is the COVID-19 vaccines. Our table illustrates many studies has been processed all over the world, and many of them are in phase 2 or phase 3 procedures. And in 2022, the COVID-19 vaccines were become the first FDA-approved COVID-19 vaccines to against COVID-19. The next one is for the hepatitis B and C. In 1992, Mason et al. Attempt to produce a vaccine plant with productions of HBsAg in the Continental bacterium, and this kind of HBsAg was successful in potato in 1997, lecture in 2011, carrot in 2002, and many kind of plants go on. One of the most interesting study was in 2020. Charlotte Zan et al. Designed a double-blind placebo-controlled clinical trial to evaluate the immunogenicity of hepatitis B surface antigen HBsAg expression in potato on 420 volunteers. Then over 70% of the volunteers giving the 3 doses of 100 dose of uncooked potato show an immune response protected against the infections with the hepatitis B virus. The next one is Ebola virus. The successful expression of 2 epitopes of EBOV glycoprotein was in tobacco in 2018, and the humanized and glyco-engineered monoclonal antibody cocktails against Ebola is produced in Nicotiana beta-methana and demonstrate a 43 to 100% survival of challenge for this kind of disease. And this kind of cocktail was approved by the WHO for Ebola disease treatment in 2014 and by FDA in 2020. The next one is for dengue virus. The oral vaccines also have been tested in humans in 2020, and edible plants such as the tomato, lecture, or banana have been selected because of their granted GRRS. Human volunteer fed on the transgenic raw potato tuber expressing the Norwalk viral capsid protein at VLPS, so a strong immune response to the antigens, due to a study of Tucker et al. 2020. The final disease for this kind of this future prospect of this kind of vaccines is seasonal influenza. The first time that seasonal influenza vaccines were studied is in 2017, and a plant-derived vaccines for seasonal influenza has also been tested in phase 3 study by the Canadian company Medicago, the newest one in 2023. That's the end of my presentation about the plant-derived vaccines. Thank you for listening.